David Sharp, co-founder and CEO of MicroCures and also a professor at Albert Einstein College of Medicine. And MicroCures was founded as a commercialization vehicle for tech coming out of my laboratory. And in particular, that technology is a new platform for siRNA-driven tissue regeneration and repair. We are, let's see. All right, we're presently a preclinical stage company with an entirely novel mechanism of action and a lot of animal data that's been supported by over $10 million of NIH funding, with, I think, very compelling results that have been published in many different peer-reviewed journals. But unfortunately, now we want to make the leap from being a preclinical stage company to a clinical one. And that's a bit of a conundrum, right? Because many investors, especially in this environment, want to fund something with human data, and we can't get that human data without the funding. And so that's something that I've been working on with Eric, and he's provided some really interesting ideas that I'll get back to later on in the talk. So an siRNA therapeutic, whoops, I won't walk away, sorry. These are small synthetic RNAs, 19 to 21 mers, that we can encapsulate in lipid nanoparticles and deliver into the cell. And once inside the cell, they'll bind to a specific target mRNA, resulting in its degradation and the temporary silencing of a particular protein. And that becomes our therapeutic target. And we are using siRNA to promote tissue repair by focusing on the microtubule cytoskeleton. Is the movie playing? Oh, God, it didn't even go. There we go. So these are dynamic arrays inside cells that I've been studying my entire career. Huh? Oh, I can walk? Thank you. Can you hear me? Okay, good. And if you look at the movie on the right, which is microtubules in a living human cell, you'll see that the array is extremely dynamic, particularly out at the cell edge, where it almost looks frenetic and dysregulated. But those behaviors are extremely tightly regulated, and subtle shifts and localized shifts in microtubule dynamics exert profound control over where, when, and how fast cells move. And we use our siRNA to exploit that by targeting, Okay, we use our siRNA to exploit that by targeting a microtubule regulatory protein called fidgetin-like 2. This is, or FL2. This is a microtubule severing protein that localizes out to the edge of the cell and normally inhibits cell movement. When we introduce siRNA, that cage on cell movement disappears, and they kick into a high gear for healing. And this is important, of course, because cell migration is a central challenge to tissue repair. Cells need to infiltrate the wound zone. Something that's really interesting about FL2 is that while it's expressed during embryogenesis, it is effectively turned off thereafter and only gets aberrantly and rapidly overexpressed after traumatic injury, where it forms a barrier to self-healing. And what we're finding is that we can use topically administered FL2 siRNA to reduce that barrier to cell healing, unlock cellular behaviors that heal, and it does so in a way in which cells don't forget who they are or what they're supposed to do. So we don't just end up with an undifferentiated mass of cells in the wound zone. They function and differentiate appropriately into the higher order structures that are important for organ function. Okay, so our lead indication, and why I'm here today, is NK. And I don't need to explain NK to any of you here, but it is a relatively high value, high growth market, presently with limited treatment options. And so we think that we can really address an unmet need, and we do that with something we call MCR231, which is effectively an FL2 siRNA eye drop. And we have a good deal of animal data to support that contention. I won't go through all of it because I don't have that much time, but just 2 slides. First, focusing on corneal healing, corneal surface healing. This shows the results from a severe corneal chemical burn done in rats, and treatment with MCR231 every other day actually dramatically enhanced the surface healing with a smooth corneal epithelium, less vascularization, less opacity, to the point that you can actually see the underlying pupil. Very, very different from controls. And simultaneously during the same time course, we saw corneal reinnervation. Now, in these injuries, the cornea really doesn't reinnervate properly, no matter how much time you give it. But with MCR 231 treatment over 10 days, we see a nice radial innervation. And quantitatively, if we look at nerve bundles, it is indistinguishable from uninjured corneas. So along with these efficacy data and with NIH support, we now have an optimized formulation with all the physiochemical properties that we'd like to see to drive it further into clinical development. So that is our next goal. And in terms of crossing the valley of death, it's been projected that if we started these studies now and did a smaller first-in-human study, we would actually be getting our first human data in a little bit over a year. Okay, so we'd be on the other side of this dreadful valley. Okay, just to conclude, we are a preclinical stage company developing siRNA therapeutics. The mechanism of action is unique. We have our first indication as neurotrophic keratitis. Lots of animal data, but no human data. And so we're squarely in the valley of death, and I look forward to continued discussions and input from Eric for how to proceed. Thank you.
I want to entertain with the two of you following the mentor-mentee program. How exactly did it work for you? What did you hone into? Maybe we'll start with you, David. What's the process you went through with Eric in thinking out of the box about crossing this valley of death?
Yeah, so Eric and I spoke weekly for about a month. He went through my slides, told me what to put in, what to cut out, and really nailed down what our problems were. You know, if we're looking to raise for a traditional phase 1/2A, which we're happy to do, that's a big ask. But what Eric suggested was a workaround in which we do a small OUS proof of concept study.
Wait. He'll tell you what he wants to do.
I'm just repeating what you told me, word for word.
You're doing great.
Yeah. Go for it.
So that was the process. Go ahead, Sensei.
Yeah. Well, that's a big statement. I think what I saw in MicroCures is what a lot of companies have is, number one, particularly MicroCures has got a platform. Platforms are great, but you got to have some focus. There's a tendency to talk a lot about all the things it can do. There are potential concerns from investors about lack of focus, so we talked a little bit about that. And then I think the biggest challenge that we faced in getting through our phase 2, which we had to really scratch and scramble to get done, was how to get the money in the door to be able to afford it. We then learned from that and decided to use an outside US approach, which saved us a significant amount of money on our phase 3, which will close up at the end of next month. And that idea of reducing the overall burden to try to get in front of people with a much more cost-reasonable proof of concept in humans was probably, I think, the biggest piece of this. Really intriguing technology. I mean, you saw the video on the screen. It's very cool. It'd be great to see it working in people. It's kind of what we went through.
I agree.
Yeah.
So in essence, you implemented some of the experiences that you've had in Stuart Therapeutics with regards to the phase 3 trials.
Right.
Into an earlier stage, trying to get the money in the door with that approach.
Yeah.
I want to say that from my perspective, MicroCures have been very fortunate, first of all, to have a first of its kind, top-notch science that was also from a preclinical perspective was awarded by several NIH grants, which actually add to the clout when you come to investors, because if NIH thought that this was warranted, then it's probably worthwhile, the look. So from that perspective, you have exhausted the NIH opportunities, right, or grant funding?
Well, I mean, we continue to have NIH grants for R&D.
Yeah.
But yes, we need to move past NIH grants to become a clinical stage company.
Yes,
this point.
absolutely, absolutely.
Yep.
So with that said, we wanted to involve two more people to have their two eyes on it. I would like to invite Rob Rothman, who is standing by the wayside. So thank you so much. And David Almeida, please come over to the stage. I'll stand up so that you can sit up.
I can roll down.
No, you're good.
Thank you. Love the shoes. Thank you.
So you two are representing two pairs of fresh eyes on the conundrum of crossing the valley of death. Rob, you're coming from an investor perspective and a physician who's involved in clinical trials. What key factors do you look for in a startup with regard to preclinical data to determine whether it's ready to leap to the clinical stage?
Well, I'm going to qualify this by saying that my eyes aren't so fresh anymore.
Specifically on MicroCures.
Well, I know MicroCures well.
Yeah.
So MicroCures is a company with science that I think is absolutely fascinating, and I think everybody who knows my involvement in the venture life cycle of ophthalmology would know that it's something that would excite me, right? That's kind of what we invest in, is exciting science. We're also, as a venture stage investor, much more willing to accept certain risks early on to try and propagate companies to get to that next level. And full disclosure, if we had a second fund, we probably would have invested in MicroCures by now. So, that's kind of been the roadblock. I don't know if anybody in the room knows, but it's been a little tough out there in the venture space for the last few years, right? I don't know. Are you guys familiar with this current climate?
They're all well-funded.
Yeah,
No, I'm just
exactly.
kidding.
So, for us, and Stuart is obviously one of our portfolio companies, so the things that we require are the things that anybody would require, right? High quality science, some proof of concept. We don't require human data for that. We need some sort of proof of concept. Quality management, solid structure, and favorable investment terms. It's kind of pretty straightforward from our perspective.
I think those are great points. I think from the trial aspect, because you have this exciting IP, the science is really good, the science is well developed. So what I usually see in trying to develop a clinical program, entering into humans for the first time, is going to be this kind of desire to, all the science already, so you're going to want to put it in as your first primary outcome measures in your first phase 1 trial, which will be a disaster and will ruin your program, and there'll be no way to come back from that. So this way, it's just, safety first, and then you start becoming methodical about what your outcome measures are, and then you start thinking about actual trial design. What you want to do is because if a patient shows up for your trial and they have neurotrophic keratitis, and let's say they screen fail, you can't lose that patient, not a phase 1, because that patient may never come around again. So you need a mechanism, whether it's a screen fail trial, what's an accessory pathway trial, whether it's a alternate dosing trial. You need something for that patient to be able to, with different eligibility, so that you can build your phase 1 program fairly quickly. And now this allows you to, your investors to calm down and everyone to kind of move on.
So Eric, from your experience, what are unexpected challenges you faced with the company going from the preclinical to the clinical stage?
I think more recently, the idea of working in Australia is extremely attractive because the discount that you were able to effect from that is a significant reduction in cost. Even though I've been around business a long, long time, you always underestimate the challenge of working halfway around the world. And that's something you just have to build in. So if you look at the rebate in Australia, it's north of 40%. That's very attractive. We were able to put together a venture debt facility to allow us to pull the cash forward, which reduced the seed capital required to get the trial started. But the net amount you get out of it after you start thinking about managing it is probably closer to 33%. So it's great, it's really good, but you don't overplan for that. And I think delays are perhaps a little bit more inevitable in that kind of environment, any outside U.S. trial, than they are in the U.S. So that's something you got to look for. Excuse me. And I think physician behavior outside the U.S. is very different than physician behavior in the U.S. So you have to know your target investigator. That's probably something that I would recommend as well.
Yeah, absolutely. Group questions. So it's for all of you. Feel free to chime in. What roles do partnerships, whether large companies, academic institutions, patient advocacy groups, advisors, where do they play differently with crossing this valley of death? If they do, I don't know.
I think advocacy groups are a huge part, Sharon, because you can think about a phase one, and we see a lot of these phase ones in rare diagnoses. You can have your whole registration for your trial through an advocacy support collaboration model right before you start, which I think you have to do.
Yeah, that's a good insight, specifically for rare diseases. Any other insights on out-of-the-box thinking with regards to partnering with the non-traditional partners for this preclinical to clinical passage?
Well, I think there's two components to that. The first is funding, right? So funding, you need to exploit whatever you can in terms of funding, and advocacy is critical at that stage. So, having people who understand and believe in what you're doing is critical for you being able to build a syndicate. When it comes to doing the clinical work, obviously you need to have people who know what they're doing, right? And, kind of the biggest mistake I think that I've seen in some companies who kind of fall in this space of going from preclinical to clinical is thinking they know everything, and it's generally not the case. And the unwillingness to bring in people who can help you with those gaps. One of the things that I guess we do is try and have a team together that understands as much of that pathway from beginning to end as we can involved to try and find the things that you miss and make suggestions and help you bring in those key components. So I think having an open mind when it comes to where your money comes from and who can help you is critical.
And I think from my perspective, sorry for butting in here, from my perspective, we're all looking for investments. And if you ask any startup, what are you looking for? We're looking for the investment. But the reason why people cannot raise investment is not one. There's multiple colors to this. Each company faces a different challenge, whether it's crossing the valley of death, that's a chicken and an egg conundrum of how to get the money, or other topics like the platform to product question. What do I present, a platform or a product? And so there's many questions to that, and that's why we brought you up on the podium. Eric, you wanted to add something?
Yeah, the only thing I'd add is there's an opportunity to do some pretty creative contracting with your vendors when you're doing trials both here in the U.S. and overseas. And the team at Stuart had a lot of experience in a number of different industries in doing these kinds of creative partnerships. So we did some of those with CROs, we did some of those with other contractors, and it was able to reduce our net cash out of pocket early on, which really helped us get through that phase two. I think that was the biggest part of what got us through the phase two, was that degree of creativity in that. The outside U.S. trial opportunity came for the phase three for us, but I think that's an underplayed aspect of how to get a startup through this very challenging set of steps that are necessary to get what we all hope for, which is a gigantic check at a later date. So that's something I'd think about.
Well, thank you very much, gentlemen. This has been really riveting. What I want to do is I want to take that last remark with regards to clinical trials and pivot into designing clinical trials of the future. And so thank you so much.
Thank you.